Good evening to all. So my name is Felix Gonzalez. I am a data scientist. I work in a lot of different places. But one of the things that may be of interest to you, I teach data science in University of Maryland, Baltimore County for master's level, and also teach a few other classes related to Python and data science. So I prepared these. So Matt you asked me about doing a short kind of like presentation on a few topics related to clustering algorithms and dimensionality reduction, which UMAP is part of. And I also grabbed one of the data sets that we were talking about last week and did apply clustering algorithms to that data set. So this is specific. So there's a lot of algorithms in machine learning and data science that you can use. So these presentations are going to be specific to unsupervised machine learning, specifically obviously applying it to the data set that I'm going to be talking in a little bit. So this presentation, as well as the Python code that I created, it's all available. And so I don't know if you guys have so this is my second meeting. Can't recall if you had a GitHub page for the group, but I put this on my personal page, so it should be available to everyone. I put the link on the chat. So the presentation is there, and this is basically the Python code, which will go in a little bit if you are interested. I think it's worth it because you can see the clustering algorithms in action while we make some changes. And I think that typically helps explain or helps people see how the clustering algorithms work. Because there's a few clustering algorithms, and I typically apply k-means, and then I also apply DBSCAN so that people can see, okay, I can see how k-means work, I can see how DBSCAN works. And typically, you can see once I start applying the algorithm, you can see what cases would you use k-means. There's specific cases that you want to use more like DBSCAN or what is called density-based clustering algorithms. So with that, let me go back to the presentation. And so, supervised machine learning is part of these clustering of these algorithms that basically you do not have to provide training to the data, or the data does not have specific labels. So typically, most people are familiar with supervised machine learning, where, for example, if you have an email, right, and there's some model running in the backend of most email systems, when you have an email coming in, you have the email system sends it to either your inbox, or sends it to the spam, or perhaps sends it to the social folder or a tab, depending on what, if you have Gmail or Yahoo. So everyone has a little bit different. But there's a machine learning model on the back end that is detecting, looking at that email that is coming in, and is deciding where to put the new email. So those machine learning algorithms typically are part of the supervised machine learning. So that model, you have to have given that model examples of what a spam email looks like, what a ham or your inbox email looks like, and then the model makes a decision based on similarity and what's called classification algorithm. So it's basically trying to determine a class of an email. So in this case, I am not providing. So unsupervised machine learning does not have the labels of the training data, right? So in the email system, you have to specify that training data that you're telling the model. You have to specify these are examples. So these are 1,000 or 100,000 emails that are spam email that somebody already kind of like went in and classified it. Then you also have, whatever amount of data you have available on examples of what looks like your inbox emails likes, or your social emails, or your promotions emails. But on supervised machine learning, you don't have that label. So you basically, these algorithms, you let the algorithm go in and find patterns in the data. So there's two main types of unsupervised machine learning algorithms. One is clustering algorithms, which we're gonna discuss in a little bit and show you some examples of the two main ones, which is k-means and DBSCAN, and then dimensionality reduction. So these two tend to create different things. So clustering algorithms, the main objective is to create groups. So it actually goes in, looks at the data, the different features that you're feeding into the algorithm, and it goes in and creates groups of data. So I think this is one of the things that it will be much easier for you to see in action. Dimensionality reduction, I don't have an actual data set where I run the dimensionality reduction, but in the idea with dimensionality reduction is that you may have a data set that have 20 features, for example. You may have 20 different columns. So sometimes these clustering algorithms, when you apply them into large data sets, like 20 features, they don't work well. So one of the methods that you want to use in those cases is to use dimensionality reduction, so that then you can apply the clustering algorithm. So there's a few main ones. So principal component analysis is one of the most used ones. So PCA. There's DSNE, which I have the next slide actually discusses then, and then UMAP is another one. But the idea is that there's some math going with each one of these dimensionality reduction algorithms, and you can project 20 features or 20 columns of data into 2 or 3 dimensions. So you're basically reducing that numbers of dimensions.
One thing, Felix, do you want questions along the way or at the end?
Sure, no. If you have questions, feel free to put them on the chat. And I forgot to use my presenter mode, so you probably were looking at this on the smaller side. So
yeah, no, presenter mode is helpful.
Yeah, so I don't have 2 screens on this computer, so if anyone has a question, feel free to interrupt me.
Yeah, well, I'll hand it over to Don. Don, you have your hand raised.
No, it's just a quick comment. So glad you're going through this. If you're interested in a place to publish data sets, Ulysses has for the DMP Petri dish, he has a Hugging Face space. Under Petri dish, we could also create a GitHub organization as well, I guess, if it doesn't exist.
Yeah, no, that's fine. I was just using, since I didn't know where to put it, I just used my own GitHub and put kind of like a repository for this group, and that's it. So any work that I do, I just post it there, and then we can decide where to put it. So it's fine with me.
And go ahead, Joe. You have a question?
Yes, I noticed on the meetup page it says what we need is a math and computational review of our algorithm. If someone could post a link to that algorithm, presumably some GitHub informational link, I would appreciate that. Thanks.
Sounds good, Joe. I'll send you a message in chat. Look out for that.
Thanks.
Okay, any other questions? Okay, I'll take that as a no. So this is just a table of the different types of algorithms that we can discuss. They're your typical algorithms, both clustering and dimensionality reduction. The algorithm names, so K-means and DBSCAN are 2 clustering algorithms. UMAP is part of the dimensionality reduction algorithms. TSNE is actually pretty similar to UMAP, but it's typically recommended because it's faster than TSNE. But I haven't read on what is the math that it does behind the scenes. But from what I've read, it's similar. It just works much faster. TSNE is known for being really, really slow. I have a lot of experience with PCA, TSNE, and little experience with UMAP. But typically it's pretty easy to apply because there's many libraries that just do the work for you, so you don't have to worry as a data scientist. And typically the output is exactly the same. You're grabbing 20 different columns or more. In some cases, thousands of columns of data if you're working in natural language processing, for example. So it's typically to have thousands of columns of data and then reducing it to 2 or 3 dimensions, which is the point of dimensionality reduction. Then with that data set that is basically just a projection, mathematical projection into 2 dimensions, higher dimensional space into 2 dimensions, then you can actually run a whole bunch of different data science algorithms, clustering, supervised machine learning, similarity ranking. So there's a lot of things that you can do now that you have that reduced data set. Okay, so a lot of these I think I've already discussed, so I'm going to jump this. So I wasn't sure which of the data sets you wanted me to do, but I mean, once you see it, I think it's much easier. And it's pretty easy to just update the notebook that I have, the Python notebook that I have.
Hey, I got one quick question.
Yep.
If I remember correctly, DBSCAN, you don't need to tell it how many clusters it makes, which is sort of the difference between that.
That is correct.
Like a K-means or a K-mer.
Yep. Well, we'll see.
That's sort of the problem is kind of like, well, how many clusters do I even have? I have no idea. And that one might let you find it versus telling a K-means algorithm I got 10 or something.
we'll get to that. There's actually methods to calculate what is called the optimal number of clusters, and I have both of them for k-means and DBSCAN. Well, DBSCAN, you don't tell it how many clusters, but you have to tell it the density value or density parameter that it uses to calculate the clusters.
All right.
But
Cool.
we'll play with it in a little bit. So I have some static versions of the data set in the next few slides, and then I'll open my Jupyter notebook, which is running in Python, and we'll be able to make changes. We'll be able to see, okay, what is the optimal number of clusters? We'll be able to put around, different clusters for k-means, different parameters for the density value. We'll see a few of the things that you can do with DBSCAN, and a few of the things that you can do with k-means. So yeah, so that is true. So the data set that I used is for these anti-cancer, or this paper discovering the anti-cancer potential of non-oncology drugs by systemic viability profiling. So I think it's based on this paper, and they also, and I can put this on the chat. And they also have, I put also this on the Slack channel. And then let me grab the other one. So they have also a, do I have two of the same presentation? Oh, come on, really? I don't know why I had two. So then the other one is a dashboard that actually they have for these specific papers. So this plot here, we were actually discussing it last week. So this is just kind of like a dynamic dashboard, and you can go in, look at the data, zoom in, zoom out. You can select a few of their different groups that they created. So you can, put in a few of them. So you can see the yellow, I think it was this one over here. So you can see I'm, kind of like turning them off. So there's a few things that you can do here to play with the data. So they basically, what I did is I downloaded the UMAP data. So I'm going to be using this plot here to actually show you how k-means works and how DBSCAN works. So this is a data set that has already been reduced from a dimensionality reduction standpoint. So you can see they already plotting the UMAP. Now, I do not know the data set that they started with. I would assume probably one of these data sets over here, or I think they published a few other CSV files here, but I would have taken me a little bit longer to figure it out. And I wanted to show you something that we can use for the discussion in terms of clustering. Really, and I have a few recommendations on things that we can pursue that wouldn't be too difficult. Okay, so this is the data set that I started with. So here is just another example. So I have these, basically I recreated the data set in my Jupyter notebook, which we'll look at in a little bit. And it's just the same thing. I just grabbed the different groups of the data. I think they basically profiled the data set that they had, and they created 25 different groups. But they was, someone actually sat down and kind of like went through the data and created those specific groups. I do not know the details on how they did it. I presume that they discussed it at some level within the paper, but I didn't actually, because it was not the purpose of the work that I was trying to do. I did not really research on how they created these groups. So when you are using, so what I did in Python, and I went in and I grabbed this data set, and then I applied k-means. So you can see how k-means basically redivides this data. And I did the same thing using DBSCAN. Now, in some cases, depending on which algorithm you're using, how many clusters you're assigning, how many what's the epsilon value or the density value that you're telling DBSCAN, there may be some overlaps between the groups that the paper has and the clusters that are going to be resulting from DBSCAN. So any questions so far before I jump into some of the demonstrations here, or some of the examples, and then we'll jump into the actual demo of Python.
If you could take a minute just on the axes, are these axes kind of, I know it's like a reduced two-dimensional representation is kind of an abstract thing. Yep. I don't know if they have meaning.
So let me just show you the raw data. It may be a little bit easier just looking at the raw data. So this is basically the raw data. So if you go to their page there that has so this one here, if you go download UMAP data, that's basically what you get. So you get this CSV file with a broad ID, and a lot of these I'm not familiar with. So the name, the dose, the screen ID, the top feature, the feature type, the drug category, MOA UMAP, the target, the disease area, indication, phase, that's by modality, coefficient, COR, I'm not sure what that one is, hub URL, the group. So these are basically the groups that you see in the, we can look at a few of these here as well. So we can apply filtering. So we can see the group here as well. So these are all the different groups that they actually plotted in the data. They have BM, which I'm not sure what it is either, and then the X and Y, it's the UMAP 1 and then UMAP 2. Now, in theory, before the raw data of the resulting data set that we're seeing here probably had some other columns, probably, and I don't know how many. In dimensionality reduction, you can have from 4 dimensions all the way up to thousands of dimensions, and it would be, you may have another column here, numerical column typically. I don't know, Z. You may have some other column, and then I don't know, whatever, QA. So you would have other columns here, and the idea with UMAP is that it's just grabbing all whatever other columns that you have and creating a projection into 2 columns. And the math is different depending if you use PCA or you use TSNE or you use UMAP. And there's other dimensionality reductions as well. So I don't know if that explains what is happening with any of the dimensionality reduction algorithms. And I may have some other work that I've done with like PCA, but I may have to search if you're interested, or I may be able to prepare something if you have another data set that has more than 2 dimensions, just to show you how these dimensionality reductions work. But basically, they're just creating 20 columns of data and converting it into 2 columns of data. The idea is that they are trying to keep the majority of the information of the other 20 columns. So they do some math in order to determine what is the new resulting 2 columns. So these X here and this Y here are the UMAPs 1 and the UMAP 2 that you see here. So when I reproduce the data, I wanted to make sure that I was grabbing the correct data, and you can see, so this is my reproduction. You can see it has different colors. I didn't really bother too much about the colors. I just wanted to see that the shape is maintained, and you can see my reproduction, it's basically the same. There we go. So you can see it keeps that same kind of like circular area here, as well as those clusters up here, and then kind of like your main cluster. Okay, so once you have this data set, and I saw that I didn't have to make any changes, I said, okay, let me now go in and apply with now my 2 dimensions, so UMAP 1 and UMAP 2. I'm going to apply the k-means clusters. So I'll show you in a little bit the plot for the optimal number of clusters. So basically what k-means does is I can tell it, okay, go in and find 5 clusters, or 4 clusters in this case. And you can see it's going in and reclustering the data. So it's creating that group there, so a purple group. This X here is the centroid. So k-means is one of the algorithms that is centroid-based. Basically what it does is it iterates through the data to calculate, depending on the number of clusters that you're specifying, it calculates the most optimal distances between 4 clusters and the rest of the neighboring points of that centroid. So you can see it basically has this cluster here, has this gray cluster here, has another cluster here, and then this other cluster here. So this is if I use k equal to 4. Now, one of the challenges with k-means is, as was mentioned in this group, yes, k-means you have to specify the number of clusters. The other challenge is that k-means will force every single data point into a cluster. So k-means, you cannot use it to identify anomalies, for example, or outliers. DBSCAN, because you're not forcing every single data point into a cluster, you will be able to find outlier data points. And most of the time you can see, okay, these may be, for example, 2 outliers because they're kind of like separated. These may be another outlier. And visually, we can go through these process where we can say, yeah, I can see if I wanted to create a cluster, perhaps this would be one cluster, this could be another. There's a whole bunch of points here that are pretty close together. So I could go through that process visually of the data set, even without this. So we could go in and use this same plot and go through that process visually and create clusters that way. But the idea with clustering algorithms is that you can determine a mathematical optimal way. If we're doing this visually, it's not a mathematical optimal way. It's kind of like estimating where are good clusters, and that's it. And in this case, they created just different groups, potentially just based on the different drugs that they were looking at. So this is what K-means does. If we go now, and I'll show you some examples where we can play as well dynamically applying the clusters. So these are, if I pick K equals to 25, so 25 clusters, you can see now it went in and created 25 different clusters. And you can see there's now, depending on how you like to see the data, there's some clusters that look like they're, much better in some cases, like this one here, like this one here. There's another cluster here. This one got divided into a whole bunch of different clusters. So basically, that's really what K-means is doing. It's going in and looking at the most optimal way to divide that distances between all the neighboring points and the center of the or the center of the cluster itself. And you can go in and divide this data as long as you have less K than data points. The algorithm is gonna go in and calculate it, right? So you have to make sure, obviously, if you apply more. If you have 1,000 data points and you give it calculate K equals to 1,001, you're gonna get an error because obviously you have more clusters than data points. So you have to be aware of things like that. Now, typically you would not do something like that. Typically, you want to be able to manage or determine a number of clusters that you can manage, and after you are done with the clustering, you can actually now select these different groups or clusters of data and look at them individually for any characteristics. And we'll talk a little bit more about that as well. Now, in DBSCAN, it's a little bit different. You have to specify the EPS value or epsilon value and the minimum samples of a minimum number of samples that you want the algorithm to put within a cluster. So we can see here I use an EPS value of 0.2. So let's see some examples here. So 0.2, if you go the UMAP 1 and the UMAP 2, and you draw what the DBSCAN is doing, it goes in, let's say it starts with this point, and it goes in and checks, okay, do I have my neighboring point? Is it in a direct distance within the 0.2 of the different coordinates? It goes in and calculates that distance. It's like, okay, no, this is not within 0.2. And goes into the next point. It says, okay, I can see this one here probably is within 0.2, but then it goes into the next neighboring point and sees that it's too far, doesn't meet the minimum samples in a cluster, and goes, okay, so this is probably going to be just outliers. So then it goes in and does that iteration from every single point in this data set. So it's looking at the neighbors and iterating and looking at, does it meet the EPS of 0.2? And then is it looking at the minimum samples in a cluster, which I define as 5? And if it does meet both of them, then it goes in and creates a cluster. So you can see this one here is a cluster, this one here is a cluster, this one here is a cluster. You can see some clusters here, part of a cluster here. I think all these red points are outliers. So they're not being assigned to that cluster. So if we go back to the 25 points, you can see, yes, there's some similarities within the cluster assignments, but DBSCAN is looking at, in this case, a titer. And we can go in and adjust this 0.2, and we can say, I want to put 0.15. And we'll do that in a little bit, and we'll see, how the cluster changes.
Joe, do you have an active question? Joe, you're muted.
Whoops, sorry about that. I was wondering about Felix's thoughts on how the null hypothesis comes into play with machine learning clustering.
So it does not apply, I think. So in null hypothesis, you have to have labels. So in this case, we don't have any labels. So we're letting the unsupervised machine learning apply the labels. So in this case, it's just the cluster numbers. So you would use null hypotheses when you're looking at accuracy of the supervised machine learning type algorithms. So in this case, it does not apply.
Well, considering that, we're using this to carry out science work, I think that it would be an unusual situation to have a statistical analysis of science work and, not make use of the null hypothesis. Namely, what are we trying to disprove? Because if we don't do that,
Yep.
then we're going to kind of end up in a situation of gradualism.
Yeah, so when you're using unsupervised machine learning, you really want to let the algorithm really just find patterns, right? And this is basically what it's doing. It's going in and saying, okay, what are the clusters that it's seeing? And you can do that on 2 dimensions. You can actually do it in all the dimensions. If we had the raw data, it's not really difficult to apply full set of dimensions. You could, if we find some patterns that we see that maybe worth exploring, I think at that point then we can say, okay, what is our hypothesis, and then we can use our null hypothesis then. But I don't think right now really we're not doing anything. We're letting literally the computer go in and find clusters.
Yes, I'm kind of pointing out that the computer can show us where to look and what to consider, but it's up to us.
Yep.
To differentiate, to make a difference between what we're trying to, disprove and a positive statement. So it's, this is like the first step.
Yeah. So let me. I think in the next slide, I may dive into where do we want to take this next. And I'll have one example. I mean, I'm not a biologist by training, and I'm pretty new to these data sets specifically, so I would have to look into the data a little bit more. But let me actually see if I think the next slide may dive a little bit into some things that we can pursue. And I think at that point,
question. So anyway, but I don't think null is the same as unknown as you're describing it, or yet to be defined label.
Yes, I kind of tripped over my words at the end there, but I think you get the gist of what I'm saying. We have to be careful not to make charts and think, we're learning something from this. We have to, make certain cognitive differences, to put it that way. But I think you get the gist of what I'm saying.
Yeah, let me. I think let's actually wait for the next 2 or 3 slides, and let's come back to the null hypotheses topic. Okay. So if we go, so a few concluding statements. So, what would be the next steps, right? So in theory, we want to look at, what are the relationship between the records? And for example, so if you go into the original data, you see that there's a kind of like a box there that I think may be worth exploring. And, as I said, I'm not a biologist, so it's really up to you on where we take this. But one example, so if we go down to the original data set, so down here, we can see there's like that cluster. And I think both DBSCAN and K-means, and depending on how you use them, they'll divide this data set. So this was part of the glucocorticoid receptor agonist. Most of them were part of that group. And then the rest of them were part of the other. So one of the things that, clustering algorithm is saying, well, all these, based on the UMAP data, and you have to be something a little bit careful with dimensionality reduction, because sometimes they really just, kind of like tend to group the data like we're seeing here, and sometimes it really doesn't mean anything. So you have to be a little bit careful with dimensionality reduction. But you can see many of these are others that are clumped together to that glucocorticoid receptor agonist. And it looks like, for whatever reason, the UMAP results do follow that for these whole bunch of them. So, and we can see these are all part of, think of the other. So we shut them down. We can see that they disappear. We bring them back up. And there was one here that is part of the, what was it? The HDAC inhibitor. Yeah, there we go. So that's the one. So that's the mechanism of action. So one thing would be, okay, is there any correlation between these glucocorticoid receptor agonists and these other type of receptors or inhibitors? So that would be one thing that would be worthwhile exploring. Now you can apply that same concept into the whole data sets and the different clusters, depending obviously on which one you use, which method you use. The clusters may be slightly different. So you can see, k-means did apply a cluster there, another cluster there, or all this was one cluster in k-means, and k equals to 4. A lot of these was just outliers based on the EPS value that I picked. But you can see both there was one cluster there, another cluster there for DBSCAN, and similar for k-means. So that would be one thing that would be perhaps worthwhile exploring. Do we have those points? And do they have any relationship? And that's typically what you want to do with. And I think at this point, then you would say, okay, my hypothesis would be, does this drugs or action mechanisms have any similarity to the glucocorticoid receptor agonist? Because my algorithm is telling me that yes, it probably has, or some similarities. So any thoughts on?
I think that you're going in the right direction, because that's something that what it is you're talking about is something that we're using anyway, and that is to use these broad categories which start to appear as you look across data sets, for just looking at them manually. You can see things like the things that I have seen, and since you're talking about it, is the glucocorticoid receptor. I know that appears in our being detected in our algorithm quite often, and specifically as the agonist. What we do not tend to see is any activity from the antagonist, which is, obviously one of the questions that you can then bring to a data set like this. If you have a clustering of agonist-like numbers, you can statistically compare them against those from the antagonist group, and they may or may not be different, as an example.
Yeah.
So Paul, I think it would be helpful to take you through some of the string stuff we've done and some of the other data sets. I think he's got the experience to de-riddle a bunch of things. So anyway, great presentation. I appreciate it.
Yeah. So a few things here. So once once we make a decision on what is the next step, what are we looking for each of the clusters, what things are we comparing, you can automate a lot of these as well. And I'll show you in a little bit. I don't know how much time I have. If you want me to dive into the Python code, and we can play a little bit with the k-means algorithm and DBSCAN and show some of the methods that you can use.
Before you do that, one of the core questions that I had goes all the way back to the beginning. And you described two columns in the original data set, X and Y.
Okay, yeah.
Okay. And from their paper and looking at all the materials that I could find associated with the paper, I had no idea of how they actually generated X and Y. I don't know what those are actually referring to.
Yeah, this is the UMAP 1 and UMAP 2 dimensions.
Yeah, but how did they get there? What are those numbers associated with? How did they generate them? I didn't see that.
Yeah, that's so as I said, I haven't read the whole paper, but typically you would have some other columns, and the UMAP algorithm. Let me see if I can find something on dimensionality reduction on my.
Yeah.
Let me take a stab.
The reason I'm particularly not concerned, but curious, is because I don't know how they took their data set and reduced it into a single or two columns, And by analogy, then I don't know how to collapse our data into 2 columns either.
Let me take a stab at that, Paul, if you don't mind. So the heart of the UMAP algorithm, as presented, it was taking multiple columns, of 20 columns, a plurality of columns, and squashing them essentially into 2 columns. And so you have this floating point value, this decimal value that as best as possible in some kind of abstract space represents all of the fields used to create those values. It's what you're doing is you're taking the raw text and the raw numbers and you're turning them into a vector. You're turning them into this kind of, you're turning like text into floating point values. And then you have a list of floating point values. And then you're collapsing that vector of floating point values into just the X and Y columns. And so you're,
I appreciate that. I'm sorry I'm being so dense, but I just don't understand what they were collapsing.
Yeah, so I agree. It is a complex concept. To me, it's kind of a form of feature engineering. That's probably a bad paraphrase, but in some contexts, you can't always just reduce it, particularly if you want only sort of 2 features. It's not like 2 individual columns or 2 individual data items. You're looking for groupings of features that logically belong together, and then kind of correlate, those 2 groupings. That's kind of my paraphrase of what I'm hearing about UMAP.
Yeah, if you guys remember, about 3 weeks ago, I had some videos on UMAPs as a basic introduction, and I guess one of the ones I didn't might be useful to illustrate it. I think I saved it in my YouTube playlist, but it kind of shows you basically how to take, it's almost like you project. They start out real simple, saying like you take like 2-dimension or a 2-dimensional map clusters. You have 2 clusters and you project that on the number line, and it kind of shows you how it organizes, how it would do it. And then they add a 3rd cluster, and they're like, well, we can sort of see this in our heads, but now imagine you have 20 of these dimensions. And I'll try to find that YouTube, and I'll put @Paul on it and post it in Slack. But it was a nice visual way to kind of see how they did, like, at least for just a few dimensions, how they reduced it.
I did look at that. So if you could put it.
Well, I think I don't think I put that on Slack yet. It was one of the ones I looked at, but I didn't put up.
All right.
But
well, I saw one that was similar. So I would be delighted.
Maybe I did put it
Michael,
up or something. Yeah.
I would be delighted to look at it. Thank you.
what I would recommend, Paul, is look at it to your intellectual curiosities. But I would recommend approaching it as a black box. It is the UMAP algorithm that is producing a similarity X and a similarity Y. And you just out of any number of columns, you specify which columns you're plugging as an input to the UMAP, and the output of the UMAP is always the two length vector of X and Y. And that just captures similarity. We don't really care how it captures similarity necessarily from like a,
Okay, I'll go with you on this one.
Yeah, I mean, it's advanced mathematics and it's computer science and data structures and
Paul, do not open the black box. Look inside.
Yes, sir.
So I do agree with dimensionality reduction algorithms. You have to be a little bit careful. I personally do not like them that much because it may send you into a black hole sometimes, or a rabbit hole. So the way that I typically, when I'm doing analysis and I am looking at dimensionality reduction, I typically do run clustering without dimensionality reduction applied. And then I go in and apply dimensionality reduction and also run the clustering just to see what I end up with. And then I try not to, one of the challenges sometimes with unsupervised machine learning is there's really no good methods to kind of evaluate how good these clusters are. You may get crap data. I mean, there's no relationship sometimes. And it happens sometimes. So you have to be very careful, and it really depends on the data. So you have to start looking at, some of these clusters that we get. Are there any characteristics? Is there anything that we want to look at from a scientific standpoint? And then kind of like try to create some automated code that looks at different characteristics for all the different clusters. I'm gonna, it's most of the cases it's unfeasible to go in through 43 clusters and look at characteristics. So it'll take weeks to do that. So you have to develop some metric based on the data set that you have. So yes, you have to be very careful with, some of these dimensionality reduction algorithms, and even clustering algorithms in general as well.
Yeah, but one of the advantages is that we have a published set with a graphic that's in principle using the same data set that we are using. So we have a role model to follow. And that's very, very appealing because even looking at the names of the clusters that were assigned in the graphic, there's enough overlap with what some of the things that we're seeing separately that it makes it quite intriguing to look at and wanting to follow up with that.
Yeah, and to build on that point, I think Felix highlighted a key challenge of any of these approaches, any of the clustering or data reduction, which is that there's no, golden truth to which clustering approach there is best. There, I'm hearing that there's a little bit of an art to narrowing in and kind of sanity checking. But what, the standard we've always used, and this is very common in biology, is at least be able to reproduce what's known. So there's this biological reproducibility standard that is it's as good as we're going to get on our approach. So like, for example, if we're running lung cancers and we know certain classes of compounds are effective in lung cancer, we should cluster to a point where at least what we do know about the biology is transparent. And I mean, we all understand it's a little bit soft. There's better and worse statistics to back up what a good fit is. But the point is, you need to make a rational justification that the way you clustered it is aligning with what can be known, with full knowledge that we probably won't ever have like a master or perfect clustering approach due to the fact that there's a lot of art involved.
Okay, so you probably were seeing me doing a little bit of bytecoding here. So just to show what is the result from, let me just restart this.
And if you could also like up the font just a bit, that would be helpful for us. Thanks, perfect.
Okay, so these just random data, 20 columns of data, all numerical. So you can see column 0, 1, 2, 3, 4. I mean, once you start, once you're a data scientist, you just see numbers. Doesn't matter if it's from biology or human resources. They're all numbers, right? So in this case, this is just an example. I'm using PCA because in this computer, I don't have a UMAP loaded. It's on my other computer. So I have, 20 columns. So 0 all the way up to column 19. Just random data, 1,000 rows of data. So if I wanted, if I wanted to plot this data in 2 dimensions, there's really no good way. I could go in, every 2 dimensions, and I will end up with a lot of, just to see one example, let's do pair plot. Let's do data. And this something takes a little bit. I don't think it should on this one here, but so these function here is gonna go in and now and create for every column, 2-column combination is gonna create its own scatter plot. So you'll see a plot for 0 and 1, 0 and 2, 0 and 3, 0 and 4, and so on for every single combination of 2 plots. So it's doing that calculation, takes a while, hopefully not too long. But then if I wanted to reduce this data, because it's known that in some cases the clustering algorithms, and in some cases, non-clustering algorithms like supervised machine learning, they may not work well with this data set. Now, this is random data, so obviously I do not know. I know that there's no relationship between this data. Most of the dimensionality reductions, they will try to find relationship within the data, but keep a mathematical representation of the features that are best correlated between each other. So you can see in this case,
to create an artificial cluster, couldn't you just like add maybe like, 5 more rows, but add 1 to all the random values. So that they should be like at least 1 off. You know, they should stand out from the rest of the random values. And
I
see where
could. I just wanted to see how does the algorithm goes in and creates 2 columns. So for example, it doesn't matter really what data there is here, right? So the algorithm, in this case, PCA, goes in and does exactly the same thing. So from those 20 columns that we have previously, now went in and created now 2 columns. So it did whatever math PCA does and transforms it into 2 components. So it's relatively easy. Now, the question is this resulting 2 dimensions reliable enough that will give us good data when we're doing clustering? And I think that's a concern from what I've seen that you have, and I agree. You have to be a little bit careful when you are applying unsupervised machine learning techniques into any data set, not only biology data, but any data set, because you may end up with clusters that mean nothing.
Does it help you to bring to it a set of data that you know is already correlated to some degree?
But and so let's actually look at the other example that I was using the data. And as I mentioned, if you go into the cluster that this created, so someone already did these clusters or these grouping based on whatever groups they created. And my guess is probably some subject matter expertise on drug interactions. So you can see that yes, there's some clusters that are relatively well defined here. And we go to the paper, the UMAP did looks like it did result in some good cluster. But you can see the glucocorticoid receptor agonist has kind of like a cluster here and then has another cluster here. So there's some good clusters that I think UMAP was able to create. And if we apply cluster k-means and DBSCAN, it does follow that same, not with 4 Ks, but if we use the 25 Ks, yeah, you can see that there's that cluster there, there's that cluster there, and there's that cluster there. Now, k-means did seem to include that yellow data points, but it also included this other group here, which seems to be, I think maybe this one. Yeah, there we go, the HMGCR inhibitor. So in some cases, k-means would go in and add other clusters from different groups that may be different. Now we can see here, this main cluster here is just a mess. So you can see it's just an overlap of a lot of different types of mechanisms. There's one here maybe for that blue, that's a HDAC inhibitor. But you can see there are a lot of overlaps here. So these are definitely significant clusters that seems to be highly correlated. Same with the ones up here and this one's here. Now, I would question then also if changing the perplexity. So every dimensionality reduction algorithm has different parameters that you can change. So UMAP and TSNE both have a perplexity, and you can go in and kind of like change those and play a little bit with those to see if mathematically it kind of like separates some of these group here. Because this group here really, when you're analyzing it, it's going to be really tough. So this one definitely, I think we're going to find some correlations. This one here. And I think if you use both the k-means or the DBSCAN, you can see it does maintain some or similarities to those data sets or to those grouping that the authors of the paper created. Now, the problem is when you start getting into this cluster here. Now, this is why I would go in and say, well, I would try to apply the clustering algorithms to the raw data without applying dimensionality reduction and see what happens.
Nathan, you had a comment on the paper. Did you want to address that?
Yeah, sorry. I just wanted to say I kind of speed read the paper here because I think that maybe this answers a question that Paul had. The information that they were using to create the UMAP, like to create the X and Y columns wasn't in that table. Essentially what it was is every think of it like a spreadsheet. The rows are the drugs and the columns are the cells. It's actually sort of like the data that we have, where it's like, you know, a triplet of drug, cell, or cell line really, and effect of the drug on the viability of that cell. So then they, you know, you wind up with a bunch of vectors that are like, you know, a given drug has high viability in cell lines A, B, C, and low in D, and drug 2 has low in A, B, C, high in F. Then you do the mathematical transformation that Felix has been describing to condense those into an X and a Y column. I mean, there's a lot more math behind it, log 2s on like how to handle missing data, but that's really the gist.
I think what's notable there is that you mentioned a triplet of cell line, compound, and response, and that's exactly what we have, right? We have, I don't know how they did their response, but we collapsed our response to a single column S prime. And you could look at, if essentially we could kind of confirm, air quotes, big air quotes, confirm our algorithm by, for example, running a cluster using the S prime as the response value in that triplet, and the other two triplets are the same. But we swapped in the S prime, and we should be able to reproduce essentially the cluster that they had. And that's kind of giving a little bit of justification that at least how our S prime is, you know, qualifying response is similar at least to how they were qualifying response. But then that opens the doors to other types of interpretations where, for example, we can look at, so that if that cluster is cell lines, plural, and compounds, plural, that also means it was like across cancers. And I would be very interested essentially to subset the cluster, and you would expect to see slightly different clusters between cancers. So for example, a lot of cancers mutate the TP53 gene. And a lot of drugs that are, you know, involving a TP53 or things like that, they would all generally be effective, but then some pathways are very cancer specific. So for example, RB1 was found in lung cancer. So we would expect that if we had two side-by-side clusters, that a number of dimensions might look the same, and then like maybe one column, like one whole batch would like disappear from one type of cluster and reappear in a different, if we're looking across two types of cancers. So and it's very interesting, things that, you know, kind of challenge us to explore what we can observe coming up.
But to Nick's point somewhat, and to yours, Matthew, you know, if we had to look at from the original data set, what could they be using, and take an educated guess as to what new number they would be using, what column number, it would probably be area under the curve.
Well, let's put it this way. That can be a literal follow-up homework. Maybe Nathan, let's see if we can find the code. I'm sure it, or I would hope it's shared somewhere. Maybe we could do some follow-up homework to see literally how did they create that clustering graphic. That might be useful for us.
Sure, I found I found the data. If I have enough time, I can try to validate the results. And
yeah, I mean,
I'm guessing that the code is out there somewhere too.
I'm assuming exactly. Just, is the code even out there? Is it runnable in some kind of sane timeline?
My recollection is there was they said there in the paper, I believe they stated there was a link someplace to the code. If I remember
I would have to imagine.
correctly.
It's such a big project that I do believe they would publish that, but we don't for sure know. That's why we need to investigate.
Oh. Yeah, one of the reasons that the area under the curve becomes interesting is because we've already done some comparisons of area under the curve and S prime. So we have that in our back pocket as a starting point, if that's what they were using, and I don't know if it is or is not.
All right. Well, we're getting stuff set up, I want to circle back to some of the approaches and also just some lateral knowledge based on ChatGPT. So we talked about clustering and dimensionality reduction, and we talked also about chaining them. So you could do a dimensionality reduction leading into clustering. That was brought up a few times. And you know, we looked at k-means, and we talked about some of the caveats being that you need to know the number of clusters, and there aren't many cases that I can think of in our data set where we have bins that were very clear on what the bins are. Perhaps, you know, there are some types of bins where that does make sense. For example, you know, if we're doing like clusters with multiple cancers and we say like this data set has 5 cancers, you know, maybe we could assume there's 5 bins. I don't know. There are probably some sub-analyses where k-means comes into play. But there was a few things I didn't like. I didn't like that you had the number of clusters. I didn't like that every point must be in a cluster, right? We talk about outliers a lot, and that's such a critical part of this data set because they're not, strictly speaking, outliers a lot of times. I think Paul and I tend to agree that a lot of times when you see a cell line doing the opposite for the same drug, like in one cell line, it's the most effective killer you've ever seen, and in the other cell line, it's like the most effective growth promoter you've ever seen. And we don't think those are outlier situations. We think those are real things that are happening. We kind of came up with like this subpopulations concept, and there's this open question about, do we have enough data to assess subpopulations? So one of the things that, you know, I'm leaning away from k-means, and that leads me into DBSCAN. Now, the DBSCAN algorithm I really liked. I think one of the things was that it had, as you guys recall, the big red dots. I'm gonna, if you don't mind, Felix, let me steal screen share for just a few minutes.
Sure.
Okay. So let me get my screen share up. So you guys recall that the red dots were these outliers, and I think that's very insightful and just a little bit more useful towards what we're doing. It comes with 2 key parameters that I could at least observe immediately, which was this EPS kind of distance value, you know, how spread out is a qualified cluster in some concept. And then min samples is actually a parameter as well, and it's actually a very relevant parameter because, for example, when we were running the DepMap data, when we were doing the pooled delta S, the pool was a pool of a set of cell lines given the context of a mutation and a cancer / tissue type. We were subsetting all of the data to those filters, and then we were also subsetting one group defined as the reference cell lines that were not mutated in one population that was mutated. And what we were observing is that the counts of cell lines that we had data for were kind of small. Like there was entire compounds where we just couldn't run the algorithm because we had like 1 or even 0 samples for one of those 2 categories. And we're just generally excluding that from our analysis. So there is sufficient data to still make large statements, but it's not exhaustive. Now, when we looked at the data that we did have enough data for, you know, what qualified enough for Paul and I when we first published the lung paper was 2. But we have at least a population of 2. Now, the min samples here is 5. So I don't know. We might have to run an analysis to see how much of our data is even qualifying for a cluster size of 5. And I think it's an important conversation to talk about the min sample size, both in the context of assuming that everything within, you know, one group or another is big enough, but also extending it into some kind of analysis of subpopulations. If you play the subpopulations card, well, guess what? You probably have to have a min samples within those subpopulations as well. So I think that's something for us to keep an eye on. But I you know, my kind of takeaway was DBSCAN was very promising. Now, the other side of this is that, as been made hopefully very crystal clear, during this call was that there is a pre-existing clustering approach on our data set by the data authors. And I think that we have to take that very seriously, because it leads us to questions like, do we need to do clustering? Do we just use this and run with it? You know, and the short answer is that this is yes, I think, but also no, because this is not the exhaustive way of clustering. It is, however, one of the most relevant ways of clustering. What is it clustering by mechanism of action? Like a MEK inhibitor is a mechanism of action. This is the most important way to cluster the compounds, but it's not the only way. For example, physical chemistry of a compound, or other clustering by gene or pathway information. That's not really captured here. So there we know that there's probably still a mandate for other types of clustering, but when it comes to the most important mechanism of action type clustering, maybe one of the things that we need to do as follow-up work is to see how far do we intend to go with this, since it's already here. For example, we probably need to at least cite this in our paper as like past and relevant context and past and relevant work. And so I think that would inform our conversation a little bit as well. But I'll yield back to Felix while I catch up my thoughts.
And one thing to add is that when you are doing clustering, sometimes you just find out things that you already knew, right? So you perhaps are not finding something new, but you're confirming, what you knew. So that's good as well. I don't see it as a failure that you didn't learn anything new, just that you confirm what you already know. So going back to the Python code, so I have these Python code here for the clustering, where I'm loading the data. So this is just the Excel. Let me go back a little bit. So this is, and I would not, I typically, running clustering algorithms is pretty easy once you have these set up. And, it's so easy that I typically just say, what, let me just run the k-means, let me run the DBSCAN, let me run the whatever dimensionality reduction I'm using. And I typically just do the comparison with all of them, because it's really easy. So there's various methods for determining the optimal number of k-means clusters. So the one I'm showing here is what is called silhouette coefficient method. It's just very easy to interpret. Is whatever are the clusters. So you got the number of clusters in the bottom, and then silhouette score at the y-axis. And then it basically goes in and iterates through all the different clusters and calculates a score. So here you have the highest one is 2 clusters, second highest one is 4 clusters, and the third highest one is 25 clusters, which were, 4 and 25 were the ones that I ended up putting on the presentation. And I can go in, I don't have to use my, the most optimal number. I can put 100 if I want, and, you'll see in a little bit. Let's see, where is that number of k? Oh, there we go. So I can change here. I can have 4. I can put 25. I can rerun this cell, and you can see all the different clusters. I can put 100. Goes in, and there we go. So we got 100 clusters now off our data set. So it's pretty easy to do once you set it up. So let's put 4. Then one thing that I typically do, so once I decide how many number of clusters I want, I go into my original data set and I extract the number of clusters. And this is just one example here. I got my data frame or my table of the different clusters for the different index. And I go in and create now a new data set or a new column. And now I have my cluster number based on k-means algorithm. And I do the same thing with DBSCAN. So now within my data set, I can go in and filter, let me grab the k-means cluster 0 and see if there's any relationship between any other thing. And I can go in and same thing. I can go into the group column and I can filter the other, or I can filter whatever other mechanism of action. So it's just another way to filter data. And you can see the count of how many records were put into each different cluster. So the idea is that once you have that, then you can look at the different characteristics of cluster 0. You can look at the different characteristics of cluster 1, and so on for each cluster.
Is
With
is
DBSCAN,
there
yep,
is
go ahead.
there is there a way to overlay two different groups? So if you run analysis 1 and then you do a parallel analysis of group 2, can you take the sum data and overlay it?
So I could, so when you say overlay, like filter for
Similarity or
I could,
identity?
yeah, I could. So similarity means a lot of things in data science. so when you say similarity based on what?
Numerical overlap.
Yeah, you could, because I could now that I have these classification or a new label, I can say, oh, let me see how many of cluster 0, what is the distribution of the mechanisms? And I can go in and do the same thing. What is the distribution for cluster 1 and the mechanism? And you'll start seeing patterns there in terms of, and I'm just using the mechanism as an example, but you can use
Sure.
any of the other columns.
Yeah,
So I
no,
can go into drug category, for example. Actually, I could even.
What prompted the question was the histogram that you were showing, where you can envision two histograms side by side arising from two different data sets, and one of the questions would be, is there a difference in the two histograms, supposing the bins are the same number?
Yeah, you could. That would be one thing that you could look into. So yeah, there's a lot of things that you can start doing now that you have the clusters, and just to show you one example.
Thank you.
So I can filter my data now for cluster 0, and I can go in and develop. Let's see, value count. So not that. Let's look at the. Let's look at the groups just to see. Let's do groups, value counts. There we go. So let's see, what did I put wrong? Oh, I know what I did. Let's see what's the name of the column. Group without an S. There we go. So for cluster 1, the majority of them are other. So I could go in and say, let me go at cluster 1. Cluster 0 was the majority of them other. But now I can see for cluster 1, MEK inhibitor, it's 14 other, and then so now I can start looking at the distribution of the different groups based on the inhibitors for the different clusters. Or I can go in and say, well, perhaps I want to look at the drug category distribution for each of the clusters. And in many cases, you may start looking at different patterns. So that's one of the things that you would start now. For proceeding after this, you would actually want to sit down as a team and be like, okay, what things can we look at between these clusters? So is it worthwhile to look at the drug category? Is it worthwhile to look at the groups? So there's some follow-up discussions on what you want to look into. So for the DBSCAN, same thing. The method that you use for determining that optimal EPS is called basically the elbow, where you basically look at the EPS value and how they change as of the samples, and your optimal number is somewhere in this elbow here. So I just picked 0.2, but we can play with it and put 0.3 or 0.015, depending on how many clusters we want to end up with. So this is the example that I run with EPS 0.2. I can go in and change it. Let's do 0.15. Let's rerun it. And you can see now that it's kind of like dividing some of these clusters into smaller ones, because I am reducing that density value. I can go in and do the same thing. Let's actually go up. And there we go. So now this one actually looks pretty similar to the k-means 2 clusters when we were using 25. But you can see a lot of these, I don't know, I think this is just one big cluster. And I think it's just reusing the red color. Something you have to be careful with, because the tool runs out of colors and then starts for the reuse colors. So, but I can actually look at the distribution down here. So in DBSCAN, it's cluster -1. It's basically the outliers. Yeah, so you can see there's 112 that are basically outliers, and there's 839 that are put in cluster 0. So I think this is just this big cluster here. of 839 records. Now in DBSCAN, typically the issue is that this big cluster really doesn't mean anything. It's just grouping everything together. That's it. But you end up with some other good clusters here that may be worth looking at. And I think this is similar to the clusters of the grouping that the authors of the paper did. So I think in this case, probably is going to confirm that the mechanism of action probably is a good grouping mechanism. And let's see where, yeah, so you can see that same clusters there. Now there's some others here that may be worthwhile looking at into why are these put into others, just different mechanisms of action. But I mean, it looks like, at least for whatever the raw data, it looks like they're pretty close, and perhaps they have some other features that they are similar enough that this is being just clustered together here, or they're pretty similar in terms of the UMAP projection. But you can see that cluster is maintained. This one here, it's divided into that small one. So in my opinion, I think both DBSCAN and K-means do give you some useful information. And a cluster like this one, perhaps K-means would be better. So I think both of them have their own merits.
All right, that's fantastic. Do you have anything else to cover on this? Otherwise, I'll move on to some next step.
No, the one thing is, yeah, as I said, we can run this, we can upload, once we run this in our data or your data, we can, add columns into your database that, specify, the cluster category that the algorithm put in. And that way you can then do filters based on the K-means cluster or the DBSCAN cluster. I mean, there's a whole bunch of things that you can do once you have the labels. But this is it, if there's no other questions.